Good morning and welcome to day 2 of the NWR Virtual Investor Conference, Series 8. I'm Andrew Gagan from Ausbiz. Great to have your company. And today we have an excellent lineup of biotechnology and medical device companies presenting. And the first presentation today comes from one of the standout performers on the ASX over the past 12 months, Imugene, ticker code IMU. Imugene is a clinical stage immuno-oncology company developing a range of new and novel immunotherapies that seek to activate the immune system of cancer patients to treat and eradicate tumours. Its unique platform technologies seek to harness the body's immune system against tumours, potentially achieving a similar or greater effect than synthetically manufactured monoclonal antibody and other immunotherapies. Just a reminder, if you would like to ask a question, please do so. You can do that by using the Q&A panel on your screen at the end of the presentation. Joining us now is Managing Director and Chief Executive of Imugene, Leslie Chong. Leslie.
Thank you, Andrew. I am delighted to talk about Imugene. Here's my disclaimer. And just a brief intro about Imugene. In 2013, Mr. Paul Hopper formed the company that we now know as Imugene. I joined in 2015. My last day at Genentech was literally September 8th. And then my first day at Imugene was September 11th of 2015 when I started. And we went right into putting our first, most advanced in the clinic, HER-Vaxx, in 2017. And we like this idea of a B cell creating your own antibody. So therefore, we in-license a whole pipeline from the Ohio State University with co-invention with Mayo Clinic on a B cell platform. And in 2019, Paul Hopper had some level of success with a company called Veralytics, and we went out to look for an oncolytic virus. And what we got was a very prolific oncolytic virus, probably the most powerful oncolytic virus now. We acquired it into the company in 2019. And then as early as this year, May of 2021, we were able to in-license a powerful product that in combination or conjunction with the cellular therapy or CAR T, you can obliterate solid tumors. So the hope is there, the novelty is there, the innovation is there. And with Imugene, you get 3 novel technology platforms: the oncolytic viral therapy, the oncolytics and cellular therapy, and the B-cell activating immunotherapies. With our B-cell technologies, we have HER-Vaxx already completing phase 2, reaping out data, as well as PD-1 vax and non-small cell lung in phase 1. And then we also have the oncolytic viral therapy, and there's 2 phase 1 clinical trials. Phase 1, phase 2, some adaptive studies happening that I'll talk about in a moment. And then oncolytics is currently in preclinical, but there's a lot of news coming off that as well. We have an incredibly experienced oncolytic viral team, as well as now we're gaining a lot of cellular therapy expertise. Significant news flow as early as a few months from now to years from now. And I am delighted to report that we are fully funded with our recent placement. All programs are fully funded such that I can focus on getting us partnered and that beautiful collaboration and collecting the data that's needed in order for that to happen. So as I said, 3 platforms. Oncolytics is a CF30, is an oncolytic virus that expresses CD19 when combined with cellular therapy. The idea here is to really obliterate solid tumors. And then the CF33 oncolytic virus, we have the ARM virus, which has a PDL1 transgene, which means it gives a direct payload to the tumor cells. And then Vaccinia, the coronavirus, we have quite a large schematic study that will start as early as this year. With Bcell Immunotherapy, our most advanced in the clinic currently, HER-Vaxx finishing out HER2, and I am HER2 studies in gastric cancer, and I am excited about what's in the horizon. So keep tuned there. And then PD-1 Vax, we're just currently in cohort 3, our highest dose, and happy to give you update as soon as we receive it. All our platform and our products have a beautiful IP that goes way out to 2038, 2037. What that means is that you have a long runway, not only to develop, but whoever takes us on as a partner or collaborator, big pharma per se, will have a long life of being able to market our products. We are led by some international team of experts that have done this before. They have experience in taking a drug from bench to bedside and making those deals. You know, I think everyone knows Dr. Axel Hoos, Dr. Jens Eichsing, Dr. Leslie Russo, who helps me lead the team, and Charles Walker as well. So I'll go into the Bcell Immunotherapy. The thing that it really excited me when I saw this platform when I was at Genentech is the way that it elicits your own immune system, your B cells, to create the antibodies. So what we have seen from that is that we've seen safety. There's limited toxicities, even when combined with chemo. We have a really nice safety profile with all our drugs under that platform. We have seen efficacy, something that had astounded us, that the polyclonal effect and also the memory effect was so nice about patients, especially in time of COVID, is that they only had to come into the clinic every 3 to 4 months just to get their booster shots. And the cost of goods, we're 1/10 of the cost of goods to make our product versus a monoclonal antibody. However, I think that will reflect in the pricing of the drug, which is nice to see. We've completed enrollment into phase 2. We've already hit the secondary endpoint of progression-free survival of 24 patients that have had a level of tumor growth. And then we're just simply waiting for the overall survival. That's the primary endpoint. And that's when we can really look at, as compared to the standard of care across our 36 patients. We're just waiting for a few more patients to get that 24 number of overall survival. We were pretty happy with the results of the study thus far and have been able to publish and present at international forums like the American Association of Cancer Research. And the data really showed us that given our method of B-cell producing antibodies, we were still able to provide efficacy with patients. And that early separation, this is a Kaplan-Meier curve in the middle there, and you want that early separation and you want it to keep separate, which is really nice. And the antibody productions were strong and high. And all patients had really high levels of antibodies, and this is exactly what we wanted to see. Again, we're just simply waiting for the overall survival so that we can collect all the data together and be able to publish it and present that. Our PD-1 Vax and non-small cell lung is currently in currently in cohort 3, enrolling patients. We're hoping to announce on the completion of that enrollment into cohort 3 so that we can start evaluating some of the data that has come through. Earlier on in cohort 1 and 2, we saw patients receive a nice response level, meaning there was a patient that had a complete response after they failed off of other lines of therapy like pembrolizumab. And I'm just delighted to think about this patient going home to tell his family he no longer has cancer. And he's had a free of cancer survival and outlook for the last 150 days or more. So I'm delighted for that patient, and we hope that we keep collecting and obtaining those kind of data. So I'll move on to our oncolytic virus. So virus, in the simplest way it works in ours for cancer, is that it infiltrates into only cancer cells, only solid tumor malignant cells. It's only attracted to this. It infiltrates, it multiplies at such an accelerated rate that it implodes or explodes the tumors, and then more virus is put onto the other cancer cell as it spreads. We have seen preclinically what's called an abscopal effect, the willingness for this virus to be able to travel. So we think that that this could work in intravenous, and that's important because currently there's no oncolytic viruses that work intravenously, only intratumorally. And you can imagine intratumorally, you have to find surface layer tumors, and that's a little bit more narrowing than going wide with intravenous. This study is in triple negative breast cancer. Lots of triple negative breast cancer patients have limited, time of survival, and I think this is a truly unmet need. We've already received FDA IND, and so we're simply waiting for the site's sort of bureaucratic ethics review board approval so that we can start dosing patients. I know that our principal investigator already has a few patients lined up, so we're just anxious to get started on this. This one in particular is unique in that it not only has an oncolytic virus, but it has a transgene of a PD-L1. A PD-L1, when you, it's already approved in some lines of triple negative breast cancer. And we think that that combination or that coordinated effort of giving that PD-L1 directly to tumors and then having it virally infected is going to work beautifully in this setting. So we took the parental-virus, the most powerful oncolytic virus that we have, our CF33, and we're putting it against many tumor types. We have head and neck here, advanced melanoma, triple negative breast, non-small cell lung, bladder, gastric, colorectal, renal, pancreatic, you name it. And what we'll do is we'll not only interrogate or look at intratumoral tumors, we'll also look at intravenous. And as soon as we've cleared 1 or 2 or maybe even 3 cohorts, we'll go right into a combination with standard of care in many different tumor types as well. So this is a pretty exciting study and pretty ambitious, but one I think we'll be quite successful at. That is destined to get an FDA IND as early as at the end of this year, and hopefully to start the study, hopefully at the end of this year or Q1 of 2022. And I'll move on to Oncolytics. So Oncolytics is this unique oncolytic virus that has a transgene of CD19. It shuttles up this flag of CD19. It veils your solid tumor in this lovely protein, and therefore a CD19 directed CAR T or a cellular therapy can come and obliterate the solid tumors. I would implore you to watch this gorgeous video by Saul Priceman and Anthony Park and Yuman Fong, our inventors. It explains it much eloquently than I have, but it's a gorgeous video and I would implore you to watch that. So as I said, there are 4 approved CD19 CAR Ts. Now, these have been garnering something like 60 to 90% curative rates in blood diseases because CD19 is only present in blood cancers. So what we're purporting to do and what we're excited about is to shroud the solid tumors in CD19. Therefore, therapies like this and other CD19 directed targets can combine with ours. So in the simplest way, again, our virus only infects tumor cells. It multiplies at an accelerated rate. It shuttles up those little red CD19 flags. When a CD19 CAR T or other cellular therapy comes to attack it, it then implodes the tumor, explodes the tumor. Really, it's just a dynamic thing that happens, and it cycles again. And my chairman has a way of looking at things quite simply that, I think it's worth repeating, in that CD19 oncolytic virus infects solid cancer cells. It puts these little CD19 flags up into the solid cancer cells and then simply implodes, and then the cancer dies. And this happens again and again and again. This therapy, the Oncolytics or CF33CD19, preclinical data was so impressive that it was on the cover of Science Translational Medicine journal. This got our attention, of course, but it also received a lot of attention from other biotech and pharma companies. So we're in many, many various different conversations that I will be delighted to share with you for when that happens. And we have, as I said, some plethora of huge milestones. These are just the sprinkling of some of the news that we'll be announcing in the next few months to years on. With checkback, we hope to get that first patient dose and then onwards from there to evaluate the various different doses and cohorts. The CD19 directed cell therapy partner, GMP manufacturer, but these are both for oncolytics. HER-Vaxx, when we reach the overall survival of 24 patients, then we'll hurriedly try to put that data together. We'd like to publish it as well as announce to the market. And some new interesting things that are happening with HER-Vaxx that I'm hoping to announce very soon. And then PD-1 vax with a maximum feasible dose and so on and so on. So that's the end of my presentation. Thank you so much for your attention.
Leslie, thanks so much for that. We do have a few questions coming your way, if you wouldn't mind. And now the first two actually relate to what you were talking about just as far as PD-1 vax. The first one being how many patients have now been dosed in cohort 3?
So cohort 3 is midway, from what I understand from the team, being completing the dose. So it's a 3+3 design. So you have 3 patients on. If one of them has what's called a dose limiting toxicity, a DLT, then you have to extend it to 6 and see how they do. And it's about this 1/3. So if 1/3 has it, then you're sort of stuck in that dose. And we haven't really had that problem. So we have 7 patients on from cohort 1 and cohort 2. And we now are in cohort 3, as I said. We really made that criteria strict because we wanted to find out exactly how our drug is working. Because we've had some level of responses in the cohort 1 and 2, we wanted to be very circumspect about cohort 3. So we're currently in, I think we've got a second or third patient currently screening. So again, I'm hoping to provide news to the market as soon as I can.
Okay, thanks for that. And just a question in relation to CheckVac. What will be the approximate timeline for the first dosing to the first clinical results?
So we hope to get first patient in as early as August. As I think alluded to, there's some red tape with the ethics review board that needs to go through, although we've already been through many, many different committees. Now that we have FDA IND, they're just checking that little list off, but their meetings are scheduled every other month or so. So once we get that, we'll start dosing patients. And really the DLT period is something like the dose limiting toxicity period and data period is like 28 days. So we'll have to get 3 patients on, wait 28 days. So maybe every, in 3 months time.
Okay. And now, of course, you referred there at the start to that recent placement raising some $90 million. So a question just in relation to that, the reasons and the use of the funds for such a large capital raising.
Look, so it was all about funding our program so that we can really focus on data collection and then the partnering activities and collaboration activities. We really want to grow across all our programs, and that's exactly what we have done. So we've funded all our programs fully such that we can now focus on collecting the data. We've also brought some aspects of the manufacturing so we'd have a lot more control over our product. Right now, we're at the mercy of scheduling, and we don't want to do that. If we're hurriedly moving through the clinic and we run out of drug, that's quite detrimental. So I wanted to relinquish that from those hurdles. So we have brought that into a contract research organization so that we have much more control. I think it's the right thing to do. And I think that's exactly what we're spending the money on, is growing our programs, getting the data, really focusing on collaboration and partnership at this point.
And Leslie, just a question in relation to your acquisition of Vaccinia. Someone wanted to know essentially what that's bringing to Imugene, just as far as any particular stage of cancer, the work that it does, and whether trials are underway there.
So the trials will be underway. So with checkback, that is basically Vaccinia is CF33 plus a transgene of PD-L1. So it's a checkpoint inhibitor, almost like a combined therapy. That's what's happening with our triple negative breast cancer, our checkback study, and then Vaccinia, which we took just the parental virus and will be hurriedly putting that in the clinic. The team is working quite rapidly on getting an FDA IND, which will likely happen at the end of this year. And then once we receive FDA IND, we can go right into start enrolling for the study. So it's a pretty ambitious study with several different solid tumors. So we're hoping at the end of this year or Q1 of 2022 to start dosing.
All right, and a very broad question, one I'm sure you're asked constantly: when can we expect you to get a product to market?
It's all about doing the right registrational study, right? You need to prove that you're better than something else, or you're significantly, overwhelmingly, you have to convince a body to be able to do that. And that's exactly what we're trying to do. We're trying to figure out what is the best registrational path for HER-Vax, what is the best registrational path for PD-1, Vax, and across all our oncolytic viruses. So it's, we are really good at that early development, putting our drug forward, doing a development that potentially a partner can take over and get it registered a lot faster than we can. Pharma certainly has a lot more legs and power to be able to put 3-4 phase 3s together in order to get it marketed. So we're just at that early stages of figuring out exactly what registrational study path would be best for across all our products.
All right, and probably a related question there: at what stage do you expect big pharma to get interested in what you're offering, particularly as far as your programs are concerned?
Well, the word interested is an interesting word, I say, in that, yes, I can confirm that big pharma has been interested, but the conversations has to be right for us. They're interested at various different times. It's according to how it fits into their pipeline, right? So it's, every different pharma company has different pipeline, different portfolio, and it's all about how we fit into their portfolio and how to develop, within their pipeline. And so it could be at phase 1. I anticipate that with Oncolytic, something like that, as powerful as that, big pharma might be interested as early as now to getting, phase 1 in just getting it into the clinic. Whereas with things like HER-Vax and PD-1 Vax, because there's so many in the landscape, they're looking for a lot more data and a lot more surety that it fits into their pipeline. But we're getting there.
Okay. And Leslie, a more specific question, just in regards to CD19, if it only targets the cells on the surface, what happens on the DNA level to the cancer cells to survive and then replicate?
Look, so it's the virus, the oncolytic virus attacks it, shuttles it, and CAR Ts generally circulate pretty much forever in your system once you're dosed. And so this happens again. There's a memory effect that happens, that you're priming your body to be able to wrong the right that has happened with the mutation, So it's your ability of your immune system to actually, you dose, and then your ability for your immune system to come and see that again and again and again. So there is an added effect that's within your own organic body to create the drugs against the mutation that's happened, or that's been allowed to happen for many, many years.
Okay, and Leslie, a question in regards to Hervax. Will it still be reported on regarding progression-free survival, or are you waiting for overall survival before making an announcement?
I think the best use of data, given that overall survival is the primary endpoint and PFS, or progression-free survival, is the secondary, I think the prudent thing to do is to wait for, we're just at a few more patients before we reach the number 24. So we have, excuse me, 36 patients on. We're just simply waiting for the 24th patient to unfortunately hit that overall survival. And then we can start looking at completely cleaning the data and reviewing the data and getting that data reviewed.
Leslie, thanks for your presentation, and thanks so much for joining us from Imugene.
Thank you, Andrew.